of introducing Dr. Lauren Ponisio. I've followed her work for many years, so I'm really excited that she came to give a talk. So, Dr. Ponisio is an associate professor in the Department of Biology at the University of Oregon, with research focused in ecology, conservation, pollinators, data science, food studies, and bees. Dr. Ponisio earned her PhD from the University of California, Berkeley, and master's and bachelor's degrees from Stanford University. Dr. Ponisio joined the University of Oregon Department of Biology in 2020, and she studies bees and their roles as pollinators in managed and natural plant communities. She's currently leading a study that aims to change how forests in the Northwest are managed, particularly post-harvest and post-fire, to the benefit of wild bees. Her research has also examined ways to encourage California almond growers to adopt bee-friendly agricultural practices, discovered how native bee species may be best equipped to survive intensive agricultural practices and climate change, and analyzed how forest fires can help maintain pollinator biodiversity. In addition to her research in biological sciences, her mission is also to promote human diversity in the sciences. Today, she'll be giving a talk titled Network Ecology of Pollinator Health, linking floral diversity, host communities, and bee parasites. Welcome, Dr. Ponisio.
And for the many other bee researchers in the room, with all that, the list of the things we've tried to do, we can all agree that, yes, it's very hard to convince people to do bee-friendly agriculture. But today, we're not going to be talking about that. We're going to be talking about bee parasites. So, but I grew up in the Central Valley of California. Many of you maybe have driven through it. We have an in and out. So this is basically the backdrop, just acres and acres of almond all blooming right around Valentine's Day. And of course, alongside those fields are tens of thousands, hundreds of thousands of honeybee colonies. And actually, growing up, this is the Fresno bee. This is like our local newspaper. I think we can all agree this is not a bee. We're not really sure what this is, but I just grew up with this understanding that people, the local economy, all of these things were dependent on pollination and particularly bees. And so I wanted to then, continue studying how to make these landscapes better for, people and wildlife. And somehow I ended up, starting a research lab. Somehow I ended up in Oregon. And we have two main goals. The first is to understand the mechanisms by which species interactions maintain species diversity. And the second is to ask how we can harness those mechanisms to then manage and restore diversity in human modified systems like almond, like timber, and all these different places. Now we have a new little lab logo, and it's a little hard to see, but on it is the most common bee in Oregon and the most rare bee in Oregon. So this is Bombus occidentalis that has a little tiny white bum, but otherwise indistinguishable. And so our general other overarching goal is to survey all sorts of species interactions and make interaction networks and describe their characteristics. That's kind of the more fun, what we enjoy doing. So breaking those two main goals down, we often approach these questions through this idea of community assembly, or often, if we've really degraded the system, reassembly. And so that's the study of the processes that shape the identity and abundance of species in an ecological community. And we focus mostly on mutualists, particularly those plant-pollinator interactions, but more and more we've been working on parasitic interactions as well, and kind of the exchange between them. And another kind of arm of that work, which I won't go into in detail in this talk, but we're also then really interested in how these interactions then contribute to vulnerability to global change. So depending on the structure of interaction networks in different places, does that make a bee community more or less able to, for example, withstand a drought, like what we experience a lot in the West these days? And bees are amazing. They're incredibly diverse, come in all sorts of shapes and sizes. There's about 700 species of wild bees in Oregon and counting. Sometimes, I'm like, come on. There's a wonderful taxonomist named Link Best, and he's always finding new species. And I'm like, oh, we have to change my talk slides again, Link. Keep the number down. But there's about 1,700 in California. It is bigger, and about 4,000 in North America. I think Utah has. What does Utah have? Terry, dim. Closer. But there's a lot of bee researchers in Utah. Okay, fine. You can beat Oregon. I guess it's okay, fine. Utah has more bees than Oregon. But really importantly, you guys know U of O. The colors are green and yellow. So we have a U of O bee. So that's really special. There's going to be a license plate. It's going to be amazing. Um, so thinking about this community reassembly and assembly, we've approached these questions mainly through looking at post-fire and also restoration. So those are kind of our main focal, maybe study systems across the West. But zooming into this mutualist and parasite interactions, bees are a really fun group to work on because they actually acquire their parasites from flowers. So there's transmission from individual to individual. So if you have an infected individual, they basically a lot of these parasites will cause bee diarrhea. And when they're visiting the flower, they'll defecate on the flower. And then the next bee will come pick it up. So, bees are basically the bathroom doorknobs of the bee world. So yeah, I'll just let you sit with that for a moment. How do you feel about all those floral meadows now, huh? So it's interesting because, through this mutualistic interaction, they can also get these parasitic interactions. And as many of you guys know, bees are in decline, pesticides, inadequate nutrition, but a really big one is disease. And I love this map because, so where it's red, that's really high pollination needs and not a lot of bees. I think there was only one county in Utah that is any color. I couldn't figure out Jim and Terry, what do they grow there? Ah, but it's also amazing because, Oregon, completely basic blank, no data. Utah, basically no data. Like a lot of the country, we actually don't know a lot about our bee communities. So just another little bit of a call to action. Apparently, Maine's doing really well. I don't go as going up there, but because it's a nice place. So thinking about these motivating theories, in understanding parasite prevalence in our bee communities, there are two that we often draw upon in wildlife ecology, and that is amplification and biodiversity dilution theories. And the prediction is that parasite prevalence will depend on both the host and the vector community characteristics. And so what does that mean for bees? So imagine you have this beautiful wildflower meadow, you have lots of the same kind of bees over and over again. They're picking up parasites from these flowers, they're defecating on the flowers, and you get this kind of like hummingbird feeder effect, right, where you get these parasites transmitting really easily between the hosts. And then to understand the dilution piece of it, there's this idea of host competence. And this has been studied a lot in like Lyme disease and other groups. And there are all these pieces that make up a really competent host. There's exposure, there's susceptibility, there's suitability, and then there's transmission. And so if you can imagine, if you have this beautiful community with all of these different kinds of bees, some of them might be really competent hosts, some of them might be not, and that'll break up the train of transmission. And so maybe, the biodiversity dilution hypothesis or theory would apply in this situation. And another fun piece of it is that we know that these parasites, these are microbial parasites, survive at different rates depending on the floral morphology. So if it doesn't get a lot of sun exposure, like the bracts of the flowers, the parasites will live a really long time. Something like a really open aster will really quickly, the UV will inactivate the parasites, whereas around the little petals and things. And so there might be another layer of this, which is, could floral diversity also reduce transmission? And also provide, diverse flower resources that maybe help the immune systems of the bees in another direction as well. So the predictions then would be, that host and floral diversity might dilute parasite prevalence, where host abundance would amplify parasitism. And then the other theory I want to talk about for this talk is this idea of, I couldn't really think of the right name for it, but it's like ecological network epidemiological theory. So the idea that the structure of species interactions, like plant parasite networks, will influence parasite transmission. So to make kind of a cute version of that, so say we have two different kinds of networks. One is very modular, so it's broken up into little compartments, whereas one is very nested, where we have a generalist hub interacting with lots of different species. And so the specialists are interacting with a subset of the generalists. So in the first situation, it's really hard to get a lot of transfer of parasites. So you're visiting, each little group is visiting its own flowers. You kind of get moderate rates of parasitism, but it's not spreading through the whole community. And on the other end of the spectrum, if you have this, especially if that generalist is infected, it's just going to pass that parasite through the whole system. And along with both of those is this idea that the more specialized you are as like at the network level, this might lead to lower transmission as well. And then at the species level, we can imagine the roles of the species themselves might affect their exposure. So if you're a really central species, so this is a species that's like really well connected in the network, a lot of different bridging pass through you, you might end up with a lot of parasites. Whereas if you're something really on the peripheral of the network, so that's like the darker colored nodes on this diagram, then you wouldn't actually get that many parasites. So in both at like the network level and the species level, we might expect an effect. And so thinking of those two predictions, we might expect at the network level, nestedness will increase parasite prevalence, whereas modularity and specialization or like selectivity would lower it. And at the species level, species centrality, so these are metrics like degrees, so how many partners you have, closeness, like how close are those other partners to you, and betweenness, how often do you act like a bridge in the network, will increase prevalence, whereas the more selective and kind of peripheral you are, you imagine you'd have lower prevalence. And so in a lot of systems for the last 9 years, we have been going out wearing cute gloves and sterile collecting bees into individual tubes. And then we, after each collection, we bleach our gloves, we bleach our net, and we recollect so we don't get cross infection of these parasites. We then take them back to the lab, either with like on dry ice or using dry shippers, keeping them really cold. We pull out their guts, which is really easy to do, like surprisingly easy to do. Just need to grab the right piece and pull, the whole gut comes out. And then we use PCR or qPCR to select for specific parasites that we know are pretty detrimental to bee populations. And so here they are. The first is a group called Cryphidia. They're these little trypanosomathids. And we know from bumblebees and honeybees that they can really impair, colony initiation. They lower foraging efficiency. They generally mess with the bee. The Varymorphas, which were previously called Nosema, are really, really nasty little microsporidians. They kind of cause like bee dysentery. They cause a lot of reduced colony size as well as adult survival. Interestingly, we never get this parasite in populations. There's only one species where we've detected it at pretty high numbers, and that's Bombus occidentalis, the western bumblebee, that is like this parasite is associated with its decline, but we basically never get it in anything else. Ascosphaera, which many of you probably work on, who work on mason bees. This is called chalkbrood. Honeybees also get it. It's a fungus. And Apocystis, which is a very generalist neogregorine, which can cause disruption of apoptose tissue and impaired colony initiation. So, general nastiness. And just a note, I'm calling these all parasites because we're catching living bees. We don't know whether they are diseased, right? We don't know if they have a disease status, whether they have symptoms. We know that you can feed these microbes to bees and get them sick, so they can cause a disease. But in general, we know they're not healthful, right? We know they're bad, so they're parasites, but we can't say like, are they pathogens in that moment? So that's why I use the word parasites. Thanks, reviewer 3. And we've done this in a variety of study systems. So we've worked in beautiful Cascade Meadows, as well as the Oregon Coast Range. We've worked in the also Cascade wildfire areas, in sunflowers in Yolo County, and the Madrean Sky Islands. And we've focused on 3 groups in particular: Bombus, so bumblebees; Apis mellifera, which is introduced, but mostly feral in these different places, except for in the Central Valley; and Melisodes, which is just adorable. And we focus on female bees because they're the ones who are doing a lot of the foraging. And yes, I know that's a male Melisodes, but look, it's a beautiful antenna. And so we've gone through all these systems collecting these bees. And so just as a off, like we've screened about 13,000 bees for all of these different parasites. One thing you'll notice, so Melisodes is in purple, Bombus is in orange, Apis is in green. You'll notice that they vary a lot in their parasite rates and also just how many we surveyed. So that's the log. Exponent of 5 is how many we actually caught. And in general, we tried to get at least 5 to 10 individuals of each species at each like survey time, but sometimes it didn't work out. You'll also notice we just don't have a lot of data from Melisodes. And so that one, it's just, we can't get them in as much of numbers except in sunflower where they are visiting in really high numbers. And we also just surveyed really different numbers of individuals in these different study systems. Some of that is funding, but some of that is also like in the Coast Range, we had tons of money, but there are just no bees there. This is the one problem of surveying the bees out there. And so for this talk, I'm just going to focus on two different places, the Cascades wildflowers and the Madrean Sky Islands, and tell you about them in detail. And then like kind of reverse out backwards and think about all the study systems together and use all the networks that we created from them to understand that network epidemiology. And then also, I'm only going to focus on Bombus for this talk because we didn't have that great of data from Melisodes, and who cares about honeybees anyways? Just kidding. But they do really different things, and so it would be you would all be overwhelmed by an excessive number of scatter plots if we did it all. But I'd be happy to chat about the APIS and other systems in other overdrinks or something. So okay, Cascades wildfires. So these wildfires were from 2020 to 2021. Most of them were in 2020. And this system combines both our post-fire interests and our restoration interests. This work has really been led by my wonderful grad student, Rose McDonald. She works so extremely hard, and our collaborator, Katie Moriarty, who works for NCASI. This is the National Council for Air and Stream Improvement, and they're like the research branch of the timber industry. And it was thankfully funded by the USDA. So thanks, USDA. Many of you guys remember 2020. It was a bummer. Everything was on fire. Probably the wind changed directions and you all got this smoke. But it was one of the most active fire years for the West Coast. It's just in time for me to move to Eugene and have a fire. So you like that middle one over there, Eugene is like right at the edge of it. So all of that it was great. But it was a bummer for people, but what does this mean for bee communities? That's what I'm sure all of you asked immediately. So bees in general really love fire. So at first, there's, open canopy, but pretty limited floral resources. Jim actually figured out that many ground nesting bees will survive the fire, but often they come out, there's not a lot there for them. But then there's this really sweet spot kind of in the middle where there's plentiful floral resources, and eventually those pesky trees come back and we kind of lose our bee communities. So there's this like wonderful time period, this nice early cereal time period where we get lots of bees. And so, landscapes are a patchwork of different fire histories. So this little area is kind of a low severity fire. So we have basically all the trees survive, right? There's kind of just a brush fire moving down in the canopy. Then these kind of patchy areas over here, this is moderate severity. So this is like, we get some tree mortality, some living trees, kind of opens up the canopy a bit, but you still have some mature trees there. And then most importantly right now for the West are these high severity fires. And this is when we have 90% or more tree mortality. So people also call them stand replacing or crown fires. And that's where we just get basically all the trees die. And so in general, we see that biodiversity, so this mix of different fire history, really promotes the diversity of bees and flowers. But in general, bees just love high severity fire. So we see higher species richness, higher interaction richness, and higher abundance in these places where we just wiped out those trees. And this was actually the picture that got me into studying bees and fire. So this is in Yosemite National Park, and it's just, right? It's just so much bee food. Look at all those beautiful, more bee food. And then, this is this just like sea of bee food. This is, I'm sure, how you all look at landscapes. But you'll notice that this is a pretty patchy, so this is all in Yosemite, and this is a pretty patchy fire. So there's only about 5% high severity in these kind of historical Western fires. Compare that to the Holiday Farm fire. So this was one of those 2020 fires, burned about 165,000 acres. Here it is over here. Eugene is like just, to the west of it. And it's about 20% or more high severity. So a really, really, really big shift. So the majority of this fire was stand replacing. And we wanted to ask, okay, so, as this high severity patch, like if we think of these high severity patches as islands, what happens as the patch size increases? So you imagine these little bees out there, enjoying their little lives, and some of them just can't make it that deep into these high severity patches. Whereas others, like maybe a bumblebee that's really large, has really strong flight capacity can, and some of them are just like, there's nothing there for me. I'm gonna stay out here. And so we wanted to ask, but on the other side of that, it is also like a giant open island. And so if bees really love high severity fire, maybe like there's going to be tons of individuals in there. If they can make it, there'll be tons of individuals in there. And so when you ask, does high severity area decrease floral and bee diversity, but potentially increase the abundance? At the same, we had this beautiful plan. And then, of course, most of Oregon, unlike a lot, unlike Yosemite National Park, where they do not do much logging, are managed forests that are managed for timber harvest. And so private forestry practices, layered on top of wildflower, will really influence the landscape. This is the winter, but it is pretty bleak out there. And so timber harvest can promote the growth of disturbance-resistant invasive plant species. Selective herbicide spray will reduce overall floral availability. This experiment was done in the Pacific Northwest and they had all different levels of herbicide, and they looked at floral availability and bees, and the more herbicide, the fewer flowers, not surprising. And if you then kind of have post-fire and salvage logging, where they remove all the harvestable material, and then replanting, this size of these clear cuts is much, much, much larger than what you'd be able to do in regular timber operations. So if you kind of like look back at that photo, these stands are at a size, these continuous clear cuts are a size that we never see usually. So we wanted to ask then, we had this control after impact, so BASI design, where we had two really large land managers, one small and one large, and we wanted to ask, can we add flowers back to these different areas to kind of like jumpstart that floral diversity again, which you wouldn't need to do in a place like Yosemite. And so what we did is we seeded into burn slash piles. So these like on the side circles are these big piles of timber residue, and they catch them on fire. And then this is what it looks like afterwards. So it's really nice because it just nukes the seed bank, lots of nitrogen, lots of carbon, great place for plants. And so we wanted to ask in general, what are the effects of these local and landscape fire characteristics, plus adding in these enhancements, which is kind of like a sort of somewhat after the fact moment, on plant pollinator parasite communities. So first we can ask, does the surrounding area of high severity fire decrease floral and bee diversity, but increase abundance? And do these relationships depend on the number of years post-enhancement? So this is only thinking about the bees and parasites, because we don't think we're really affecting the stand level floral diversity, but we were wondering, if we add flowers to these areas, can we affect the bees and parasites? And then, how does that then feed down to thinking about, does bee abundance in these systems amplify parasitism while bee diversity and floral diversity dilute parasitism? And maybe, there could be a direct effect of this high severity fire area and enhancements on bee parasitism. And in general, I kind of didn't go into it. We always expect the flowering plant community to directly affect the bee community. So the more diverse the flowers, the more diverse the bees, more abundant the flowers, more abundant the bees. I won't focus on that for this talk, but if it doesn't happen in your data, you're like, what did I do wrong? But 99% of the time, we see that relationship. And we're going to be focusing again on just on bumblebees. And I've gotten really into causal inference frameworks, read the books. So that starts with building directed acyclic graphs. They're called DAGs. Then you do all these various magical checks. And now you can build this model in BRMS, in R, and just hit run and run them all concurrently. So you have this beautiful kind of like multi-layered model where you can estimate both the direct and indirect effects. Very into it. Still seems like magic. Might need to read the causal inference book again. But but it's really fun. So they're all fit using Bayesian model fitting tools. So I'll report what percentage of the posterior is above or below 0. Okay, so does the surrounding high severity area lower floral diversity? Interestingly, no strong evidence of an effect of high severity fire. I didn't want to put overwhelming numbers of scatter plots in this talk, so I'm going to leave those for the parasite analyses and just kind of summarize the bees and plants. Instead, these like larger scale things like growing degree days and cumulative precipitation, landowner, proxy for management intensity, and elevation really determine the plant community. So in this very modified landscape, I wouldn't expect this to happen in something more, maybe in a more natural, really high severity fire didn't matter much. So then thinking about the bees, we did see actually that high severity fire area increased both bee diversity, both rarefied richness, chance diversity, and substance diversity. So like all the facets of diversity and abundance. So bees just love high severity fire. Doesn't matter how big it is. They're just into it. We didn't see an effect of years post enhancement, so putting those burn slash piles and enhancing them on the bees in the stands. We did see an effect in the enhancements themselves, but it just doesn't spill over past those enhancements. But then, the cool parasite question. So do we see bee abundance amplifying parasitism while bee diversity dilutes it? And this is focusing on Cryptidia. And we do indeed see in bumblebees a really beautiful dilution effect. Uh, though in terms of, maybe it's kind of nice, you always like things that don't amplify parasitism. We don't see much an effect of bee abundance on amplification. So we see that really nice dilution effect. And we don't see an effect of floral diversity though. So it's all the host diversity driving this. However, let's switch over to Apocystis, our other main parasite. Now we actually see a positive effect. So biodiversity amplification effect and nothing again with bee abundance. So kind of opposing effects and also no effect of floral diversity. So opposing effects between Apocystis and Cryptidia. And I'm not going to talk about Nosema / Varymorpha because we never got enough positives to fit any kind of model. And same with Ascosphaera in bumblebees. It's really not that common in bumblebees. We see it in other groups, but so we're just going to focus on those two, Cryptidia and Apocystis. And so then, is there a direct effect of high severity fire and or enhancements on parasitism? No, not really, no. So there were some kind of like really subtle interactions where, there's some like high severity area times enhancement might dilute parasitism just a smidgen more. But when you actually look at the slope of the line, it seems like the effect sizes are really quite small. So maybe it's like significant, but not potentially biologically significant. So going back to our beautiful schematic of the way we thought the world works, unfortunately, no evidence of an effect of high severity fire on the floral community. We did see positive effects across the board on the bees, but no effect of those years post enhancement. So our restoration wasn't doing much at the landscape scale. We did see that bee diversity dilutes parasitism in Cryptidia, but amplifies in Apocystis, and limited effect of a direct effect of high severity fire or enhancements on parasite prevalence. So fixing our schematic, all of those go away. That one actually flips. So, high severity fire is actually increasing species diversity, no effects of abundance, and years post enhancement didn't matter much. So, species diversity actually kind of has these opposing, like sometimes it is, sometimes it isn't, helping things out. Why might that be? So first I want to talk about the enhancements. So this is what they look like when they're nice and mature, all these beautiful flowers. In the background, you'll see there's also a lot of flowers. That's mostly what's called oxeye daisy. Bees don't really like it. It's an invasive plant that actually eventually kind of disappeared, but we have all these beautiful native plants, lots of bees in them. And what's really interesting is like in those enhancements, we see that bee richness and diversity is much, much higher with years post restoration. Abundance doesn't really change. Seems like we see this a lot where, the species that show up really varies with restoration, but, there's a lot of generalists that kind of get there and be abundant early on. We'll have year 3 post enhancement soon, but these are really super small compared to like the size of these stands. Like these are tiny. And so it doesn't, in retrospect, it's like, okay, it makes sense. These don't really affect parasitism. There's the stand, right? Those are those little burn piles. They can be the size of like cars or buses, but compared to this huge landscape, they're a bit of a drop in the bucket. And so how can host diversity dilute parasitism in one parasite and amplify it in the other? And so I actually typed this out because I wanted to get it just perfectly right. And so this could be all about this competent host dynamics. And so Cryptidia seems like transmission is probably dominated by bumblebees. And we know that Cryptidia is a parasite that's just really pervasive in bumblebee populations. And so then having higher diversity, then again, different non-competent hosts might lead to that dilution effect. Whereas Apocystis, and we also know this, infects a lot of different kinds of hosts. So we saw it in really high amounts in honeybees. We saw it in really high levels in Melisodes and a bunch of different groups. And so actually having more diverse hosts, if they're all competent, is kind of more doing that amplification. So that's how we could get the same things going on in the same place. And so unsurprisingly, parasites have different life histories. And you can also see that here. So you can see that uh, in terms of apocystis, we kind of see it like kind of much more across the board. We see really high rates in Melissodes and things. Whereas with Cryptidia, I mean, we see it in Apis, we see it in Melissodes a bit, but it is mostly in Bombus. So that even comes out in the data. So yeah, so parasites are different. We shouldn't lump them together. Oops. We'll touch on that later. Okay, so moving on from the beautiful Cascades, let's talk about the beautiful Madrean Sky Islands. So now we're scooching over to New Mexico and Arizona. No more no more timber harvest, no more wildfires, just gorgeous, frolicking, high elevation meadows. This work is done in collaboration with Shalini Jha and led by Nicole Martinez Llaurador, my wonderful grad student. Terry was involved at one time. We roped him in, but and it was funded by the NSF. So here's what one of those beautiful meadows look like. Classic Rocky Mountain meadow, and lots of diverse flowers. And we have this biogeographic gradient from the Rockies down to the Sierra Madre. And these little mountains, these greener areas are like little islands. You see that there's also this kind of rim. So there's the Rockies up there, and then there's this kind of secondary rim. And then you see the Sierra Madre down in Mexico. And so we have this kind of like stepping stone latitudinal spatial gradient that's natural. What's really interesting is people have found that, imagine these little bumblebees are chilly and they're all cuddling together, that at higher latitudes, while it's colder, bees spend more time in colonies and you see more like bee to bee transmission within a colony. And so we might imagine that this latitudinal gradient could directly affect parasite prevalence. Additionally, we have the North American monsoon, which unfortunately, because usually when these photos are from NOAA, because whenever a monsoon was hitting us, we got some really good ones, really good storms hitting those meadows. We were usually running because we were carrying lightning rods, those nets. But the different areas in the U.S., particularly New Mexico and Arizona, get most of its rain as these really beautiful summer rainstorms. And so we've surveyed these areas 2012, 2017, 18, 2021, and 2022. The first I did as like a grad student living in my car. And we see this real seasonal cycle in the monsoons. This is again from the NOAA website, where we have really high variability in the amount of rainfall. And that rainfall mostly falls between July and September. And so we get this really abrupt seasonal, within the summer cycle. And so we have this really cool natural temporal gradient. And there was a study done out in the East that showed actually through the season, we get sort of an accumulation of parasites. So as more and more bees, the idea being more and more bees are depositing these parasites in flowers, more are picking them up. And through the season, we get this increase. And so we have these two natural gradients, like a spatial gradient and a temporal gradient. And we want to understand how that like feeds down to the parasite prevalence. A cool other thing is that there was a meta-analysis done looking at the biodiversity dilution effect. And they found that if you have a biodiversity loss gradient, so those points I outlined in pink, you tend to get a consistent negative effect, so a dilution effect of biodiversity. Whereas if you have a natural gradient that's not driven by biodiversity loss, you tend to just see things that are all over the place. So that's why those like the other bars are just kind of like some of them are above the line, some of them are below the line. And so but there's actually, I don't think this is the only study of a natural gradient with bees to ask, does this also hold? This is across like all wildlife. And so how do natural spatial and temporal gradients affect the host plant community and parasite prevalence? So we have this latitudinal gradient and this monsoonal gradient. So breaking that down, we have latitude, how does host and floral diversity decline with latitude? How does that feed then from the flowering community to the bee community? Then are host and floral diversity tied to the intra and interannual monsoonal cycles? Does bee abundance amplify parasitism while bee and floral diversity dilute parasitism? Same question as before. And then is there a direct effect of latitude on parasitism? And does parasite prevalence accumulate through the season? So those are kind of the different, like very similar structure. So I'm only going to cover these two projects, but in all of these different places, we've tried to think about what controls host and floral diversity and how that might feed down to parasitism. So thinking about both the host and the bees, how does latitude and these monsoonal temperature gradients or precipitation changes affect plant and bee communities? We see an evidence of a decline in floral diversity with latitude. So the highest diversity is in the Rockies, and it gets lower and lower and lower as we move towards Mexico. Makes sense. These are Rocky Mountain meadows. So the further away we get from the source pool, the fewer flowers we get. We didn't actually see a direct effect on the bees, but we saw an indirect effect on the bees through the plants. So the plant diversity was really, really related to the bee diversity, but there was no direct effect of latitude. And unsurprisingly, we saw really strong intra and interannual climatic effects on the bee and plant communities. So they were really following those monsoonal cycles. So then thinking about how does floral diversity and bee diversity dilute parasitism? So the blue curves are where we're at that kind of like p-value of 0.05 significance level. And we see again that, well, this is with floral diversity, we see that Cryphidia prevalence has a dilution effect. Whereas, of course, because Apocystis likes to do exactly the opposite of Cryphidia always, we see an amplification effect of floral diversity in Apocystis. We see that nice same dilution effect of Cryphidia with host diversity, when we see no effect this time in Apocystis. So some similarities and some differences from the other system. Then we think, does bombus abundance, so we're focusing really on the bumblebees this time, amplify parasitism? We see a very clear increase with higher abundances of bumblebees. And of course, not so much in the Apocystis, which follows that kind of host competence hypothesis. And then, does latitude amplify parasitism, as well as does parasite prevalence accumulate through the season? We don't see much an effect on Cryphidia, whereas we do actually see amplification in Apocystis. And I don't really have much of a hypothesis of why this might be. I would have expected both parasites to be affected similarly if bees are spending more time in colonies. So if you all have some ideas. And we actually saw that parasite prevalence accumulates through the season, whereas Apocystis prevalence declines. So they just like to do exactly the opposite in every single way. So thinking back at that schematic, we saw that floral diversity declines with latitude, and we found a strong relationship between the plants and the flowers, that the host and floral diversity are tied to these interannual and annual monsoonal cycles, that host and floral diversity diluted Cryphidia prevalence, but floral diversity actually amplified Cryphidia prevalence, or Apocystis prevalence, and then bombus abundance amplified Cryphidia prevalence, but did not have a strong effect on Apocystis. You can see why our whole lab is just like, oh, if only they would be consistent sometimes. Whereas Apocystis prevalence increases with increasing latitude, Cryphidia does not. And then always again, the opposite, Cryphidia prevalence accumulates through the season, Apocystis does not. And this is where I had the moment of like, gosh, I sure wish things were more consistent in bees. And I've had moments of like, did we just not survey enough? And I'm like, gosh, we surveyed a lot of, this is many thousands of bees. So what could be going on here? So, and we've done the same analyses just over and over again in all these different systems. And so the take homes might be that, just natural species turnover and species loss, which is usually human induced, definitely are having effects on the hosts and the parasites. So we're like, okay, so that's consistent. Amplification and dilution effects are both parasite and host specific. So I didn't want to get into the honeybees, but you can also imagine another ton of scatter plots all about the honeybees. And of course they do the opposite thing as the bumblebees. So they're doing their own, everything's being influenced differently. And so what does this actually mean for our ability to predict any parasite dynamics in these systems? And so I think, in general, I think it really calls this importance of actually studying multiple parasites and hosts individually and avoid kind of lumping them into parasitism, which, bad job, Lauren of the past. You shouldn't have done that. But, when we were all starting out, I think we didn't really recognize these really big, deep life history differences between the different parasites. Yes, I'm throwing myself under the bus. I lumped them in this paper. I'll never do it again. But at the same time, I think by studying these at the community ecology level, we can understand transmission dynamics a bit more. And then, further delve into how these different landscapes are affecting them. So then zooming out to thinking back at about the networks. So maybe, even though we see all these kind of crazy things going on between these projects, when we're talking about these really specific host communities and these really specific parasites, maybe like if we just scale it back and just think of networks and the network structure, we'll see some interesting things. So reminding you of these different theories. So we think that network structure might affect community prevalence, specifically nestedness will increase prevalence where modularity and specialization will lower it. And then with species roles, we think that being like really central in the network, I mean, you have a lot of parasites, whereas being really peripheral, being really specialized might not. So, some more curves. So we have nestedness, then modularity and selectivity. So that's like specialization and selectivity. And we see, just as we expected, that if you combine, we ended up with, I think, over almost 900 different community level networks across all of these projects. We see this really nice increase in parasite prevalence with communities with higher nestedness. You'll notice that the specialization is the opposite of what we thought, but you could also imagine that if you're being more specialized or more selective on the flowers that happen to have a lot of parasites, this could lead to this pattern. And we see no effect of modularity, and this is Cryptidia. And so potentially, if there's this like shared core, think of that nestedness roles, there's this shared core of flowers that likely act as high traffic hubs for transmission. The more reuse of the same flowers, the more exposure. This likely affects that bombus are probably clustering on a smaller subset of plants. I did some other little analyses that show that. And so this repeated conspecific visits might amplify parasitism, even though the whole network looks specialized. And then it seems like Cryptidia is mainly driven by bombus, so that's why we don't see this modularity. So bombus use hub flowers that maybe are cutting across modules, and so that's why we don't see this effect. So take a guess, Apocystis does something completely different. So we only see a slightly negative effect of modularity on Apocystis prevalence. And so, maybe there's just risk of parasite prevalence may depend less on like hub sharing, like with Cryptidia, and more on whether the network is partitioned in this case. And this does kind of go with that. Apocystis is less host specific. And so if you have a more modular network, maybe your competent hosts end up in that one modular. Thinking now about those species level roles. So we're going to look at betweenness centrality. So this is bridging species. Closeness centrality. This is the shortest paths between species. Degree centrality, which is just partner richness. And D prime, which is a metric of specialization or selectivity. It's really similar to the network level version of it. And again, the solid lines are where we see strong support. We don't see much in terms of betweenness centrality. It looks like there's quite a negative slope, but there's a lot of variability, so it ends up not fitting that 97.25% threshold. But we do see positive effects of the other metrics. You'll notice again that D, that's the selectivity metric, is going the opposite direction as we thought. So the more selective you are, the higher parasite prevalence. And so this suggests that Cryptidia is really this like contact rate embeddedness parasites, species that interact widely and sit deep within the network, face higher exposure. And you've guessed it, Apocystis is absolutely the opposite. It just kind of nothing seems to matter. So maybe Apocystis is driven by environmental factors or more of these, like if we understood more about what hosts are really competent for it, we could understand better. Whereas Cryptidia seems to be, following the bumblebees, following these different predictions. But yeah, now I'm kind of just like, I need to learn more about Apocystis and what it's all about. So, across the board and all these different focal species, we see that amplification and dilution and network structure both are both parasite and host specific. And I won't get into both what we saw in Apis and Melisodes, but it was really similar. So it's sort of like there's this interaction between hosts and parasites across the board. So hopefully, we've learned some things about this piece of our little schematic, that interaction between mutualists and parasites, and how this whole process of community assembly then shapes these different transmission dynamics. I'm not going to talk about this so much, but I just want to highlight we got an NSF grant and we got to keep it. So that's really big news. That's all about looking at these interactions and vulnerability to global change. There was two transects done in California in the 1960s by this guy named Andy Moldenke, and he would go to an island to an inland mountain peak. And we've gone back. We did one in Southern California, which is pictured here, and then in Northern California. And we're going to go back and we're going to repeat all these transects. We've already done a couple years of it. I also had this dream of getting his collection, which ended up mostly in the Cal Academy of Sciences, and doing the seep the bees in extraction buffer and get their parasites. But NSF gently told us that that was a little bit too expensive right now. So we're gonna wait for the next round of grant to do that. But it would be just so interesting to look at how the parasites have changed through time. We can only really do it with Nosema because it's a spore, and so it really holds up. A lot of these others will probably degrade. But that's kind of my next dream. And this is being led by my wonderful grad student Alejandro Satijana Fernandez. And I just want to really quickly thank all of the absolutely amazing grad students and techs and postdocs who led this work. I've just been so lucky to work with a real wide array of people while at UCR and at University of California, Riverside, and at the University of Oregon. And all the wonderful taxonomists who have helped me identify these bees. I actually made Link best like put on a lab coat and identify frozen bees under the scope. He's not he's still not happy about it. He hasn't recovered. But you'll see Terry pictured here. He's over there. He helped a lot of with the biodiversity experiment. And I've never found a picture of Joel Gardner. Yeah, can't find him. He has no web presence. So if you guys ever know what he looks like, thanks to him. And here's my to leave up on the board while you guys ask me questions, summary of all of our main results. And thank you all so much for inviting me here. I've had such a lovely time already, and I'm excited that it's 2 days long, so I get to meet with even more people. And if anyone understands this Apocystis criteria thing, please let me know, because it keeps me up at night. Yes. But thank you so much. Yeah. Brian.
Good thought. I was curious if what data you have with co-occurrence of these two parasites and whether or not it seems like there's a lot of different hypotheses that could explain why you're seeing opposite. Well, but there might be some kind of selection for reduced competition with these two parasites.
So as in geographic co-occurrence or within a host co-occurrence? Mm-hmm. Yeah. Most of our bees only have one. Only once did we get really high levels of multiple infections, and that was in the Sunflower Yolo County, and that was mostly in the managed honeybees. And we had an individual with all 5 parasites. Yeah, poor little bee. She probably didn't make it very long. But we tend to usually see, and there might be, the hard part about only catching living flying bees is they're healthy enough to be out flying. So it might be why we only see the one infection. But I could imagine there being some, either founder effects or things going on where that's why you only get the one infection. Geographically, these are very, very widespread parasites. So I mean, Crythidia in particular has been studied in a lot of detail from labs out in Europe. They seem to just be endemic in these populations. I don't think we definitely see fluctuations in how much prevalence, but we've never sampled any of these populations that don't have them. And we tend to get 3 main species. We just kind of lumped them together. We get, it's called Crythidia bombi, Crythidia expoeckii, and then Crythidia, it starts with an M, and it's mostly in honeybees. So that one, and we used to, so when we do, this is a multiplex PCR, and we get nice bands for SPA and then the 2 different species, and then we would send off the SPA bands and get them sequenced. That eventually got very, very expensive, as you imagine, $8 each to Sanger sequence them. So, but in general, we saw a good amount of co-occurrence of expoeckii and bombi even. So it's not like we only saw expoeckii, even within the species level, they seem to just kind of be everywhere. And same with Apocystis. And that one is really broad hosts across, across honeybees, bumblebees, bunch of ape, the whole that whole family of bees. We don't see them so much in the like the megacylids, like a lot of the more distantly related species. So, which makes sense. Yeah, so there's a lot more host specificity than I think well, I'm sure actually like a bee parasite person would be like, yeah, you ecologists, we knew that. And we're like, oops, like, I guess we shouldn't have been them, like, but, but I think that it's only been in the last maybe like 15 years that people have been really doing like this broad scale surveys of natural populations, whereas they're usually studied in the lab. Yeah. That was a very long answer to that question. Sorry, Brian. Yeah. Yeah, Jim.
When I first be a flower burn,
Mm-hmm.
that saw no pattern in those flowers that they visit.
Mm-hmm.
I was wondering for bumblebee flowers in Sky Island, these all in the Floral University, are those all species of bumblebees?
Ooh, so yeah, we were talking, so in the harvested forest system, we at first didn't, when we were analyzing the data, we didn't see a difference between the enhancements and the stands. And we were just like, what? Like all of that work and didn't make a difference. And then we dropped all the flowering species that bees never ever visited, like in the, you know, we started surveying in 2020, so or 2020, 2021, everything was on fire in 2020. So, 4-ish years, field seasons are 5, I forget. And we've never saw that visiting. And so when we actually did that, we saw really, really big differences. In terms of floral diversity, I think we left floral diversity as any of the flowers we saw any bees visit, not specifically bombus, but it might be good to try it both ways. I mean, bombus, as you know, visit many, they are quite generalized, but there's probably some species they won't touch. Yeah, that might be interesting to try both ways.
The other question, you talk about ultraviolet radiant rather quickly, radiant. I wonder if sunlight has a relationship with susceptibility. Bumblebees, because it's so many tubular flowers and pendant flowers like vaccine. Right? Whereas little short tongue bees are mostly walking. Walking around on rather shallow flowers.
Yeah, and we don't know that much about host competence, but one thing we do know is that larger bees, even bumblebee individuals within the same species, have a higher parasite load. So this is like qPCR. Lynn Adler's done a lot of this work. So they actually qPCR'd the gut and looked at, okay, if we can all hope that, copies of DNA correlates to infection intensity, which we're not entirely sure about, but the larger the bee, maybe it's basically like the larger the habitat size, then they have more parasite loads. And they also, as we know, disperse more. So they might be picking up parasites from other places. And I think that body size is generally correlated with tongue length, generally. I mean, I know that bumblebees can vary somewhat with tongue length and body size. That would be nice. We have some carpenter bees surveyed, but they tend to be in really low abundances. So I know the solitary bees have been hard to figure out just because, we had a couple focal groups that we wanted. We're just like, okay, if they're an Andrena, like one of the early, I feel like I'm just talking deeply bee talk right now. There's a couple of focal groups of solitary bees we really, really wanted, but then when it came down to it, we'd get like, 5 individuals of a species in a place. And, what are you going to do with that? And so we actually have a lot of just really sporadic data of these different other solitary bee groups. We've just never had enough to be able to actually fit a model, beside the Melisodes to some extent. But even the Melisodes, like, we never got such good numbers like we do with Apis and Bombus. So I don't know how I mean, more intense sampling and more bee murders. I don't know. Yeah.
So think about an earlier slide you showed some of these parasites can affect foraging behavior and they can affect the selectivity. So when you're thinking about network structure shaping prevalence, which makes sense, is it also possible that some of these parasites are shaping network structure, altering the sunflowers.
That is super interesting. I don't think anyone's thought about that. It would be really fun to experiment with. Maybe like in a greenhouse where you like inoculated flowers. Oof, that'd be neat. Yeah. Yeah, and the studies done on the floral morphology were done that way, where they inoculated different parts of flowers, and then they actually looked to see if they could get replicating parasites off the flowers again afterwards. And that's the one that saw those. Yeah, so it's possible to do. Yeah, Miranda. Yes. Mhm. Yep. Mhm.
Do you have any thoughts on like how these parasite dynamics like kind of affect? I don't know, like what do you think of the nutritional diversity aspect of this type of work? Have you looked at that?
Yes, and we looked at it through the proxy of floral diversity. Because nutritional diversity is really tough to study in wild populations. A lot of, if you imagine like trying to like get all the pollen off some flowers, I mean, you need a lot of flowers, and then you've really messed up your natural community. We had this dream too of like doing nectar and things like that, and really quickly realized that just it in a natural experiment, it's too difficult to measure in an accurate way. But thinking about that floral diversity, there could be this dilution effect, but there could also just be like, bees are healthier here. There's more nutritional, we know different flowers have different nutrition. And so there's more nutritional diversity out there, and maybe that's providing something. And so, and we did see that effect in the Sky Islands. We just didn't see it so much in the harvested forest landscape, where admittedly, floral diversity is much lower. Like the spec, if I like lined those two graphs up, like the whole axis would shift down from these beautiful high elevation meadows. So it could be this, we never hit that threshold of having enough floral diversity to matter. But in the paper where we admittedly binned all the parasites, we saw the same nutritional effect of floral diversity so when there were hedgerows next to these sunflower fields, we saw lower parasite prevalence. So hypothetically, from maybe nutritional diversity. Yeah. Yeah. But that was, we binned them. It was mostly apocystis in the Melissodes. I've since reanalyzed the data in a moment of panic, So, yes. Oh, did you raise your hand? No? Nope. Oh, yes. No. Oh, scratching. Okay. No. Do you have any big questions about it? No. Awesome. Well, thank you so much.